Okay, can people hear me? There are some problems with the webcast again, but that won't affect you guys, but it'll affect you if you want to come back and watch this lecture later. I'm not sure. They may have a webcast today. I don't know, okay? I'm going on. So that's why I'm putting things on the board that I had not planned to put on the board. Check out this board today. Number one, I'll have office hours today after class, but I will also have office hours tomorrow from 9 to 10. It's a wonderful opportunity for you to get your questions answered because there are far fewer people than the 30 or so who are coming to office hours after lecture. And this is the schedule for starting Friday through next week. Briefly, today I'm going to talk about oxidative phosphorylation, finishing up respiration. On Friday, I'm going to talk about the so-called light reactions of photosynthesis. And on Monday, I'm going to talk about the carbon fixation reactions in photosynthesis. I'll have office hours Monday after lecture. I will have a review Monday evening covering all the material. That review will be strictly questions and answers. If there are no questions, we're finished in 5 minutes, 10 minutes, never happens. That will be from 5 to 7 in 2050 VLSB. 5 to 7. No, sorry, 7 to 9. You got something else to do from 5 to 7. 7 to 9. Wednesday, Mike Meehan is going to be here from 8 to 9 to answer any last-minute questions you might have concerning the midterm. Friday will be the midterm, number 1, and you'll get a handout about that as to where you should go. So it's a little complicated. I, unfortunately, will not be around after Monday night. I may reappear magically Friday morning in Pimentel for the groups that are here, but I won't be around Tuesday, Wednesday, or Thursday for sure. Okay, this is a very brief review of what I was talking about on Wednesday. I went through a lot of stuff on Wednesday, and it's all very important, of course. So what we're talking about is respiration. Glucose plus oxygen being converted to CO2 and water. But I've modified that equation because one of the major functions of respiration is to make ATP. So now I have added XADP and XPI being converted into YATP. And instead of holding you in suspense, I will put numbers here. This number will turn out to be approximately 38. This number approximately 38. And over here, of course, 38. So you can see how much ATP is synthesized during respiration. What I talked about on Monday was glycolysis, which produces pyruvate, 2 NADHs, 2 ATPs by substrate level phosphorylation. And I emphasized one reaction in glycolysis, reaction involving glyceraldehyde 3-phosphate dehydrogenase, and I expect you to really understand what's going on in that reaction. Then we have pyruvate oxidation and the Krebs cycle, which produce CO2, lots of CO2. 2 ATPs produced in the this occurs in the cytosol. 2 ATPs produced by substrate level phosphorylation in 6 NADHs and 2 FADH2. These reactions occur in the mitochondrion. Some of them, one reaction in the Krebs cycle that we'll talk about more today, is actually in the inner membrane of the mitochondria. So after this sequence of reactions, we have fully oxidized glucose to CO2. We've made a very small amount of ATP, and there is no direct involvement of oxygen in any of these reactions. So we're in a position now where the cell has a lot of NADH, has a lot of FADH2, it's got NADH here, it's got NADH there, and just the way I talked about glycolysis, you have to regenerate NAD from NADH for the reactions of glycolysis to occur. That's certainly true here as well. You have to regenerate NAD, so glycolysis will occur. But you have to regenerate NAD and FAD in order for the Krebs cycle to occur. This happens in a series of reactions which are known as oxidative phosphorylation. What happens in a very general way, and I'll go through the details, is that NADH and FADH2 are oxidized by molecular oxygen in a complicated series of reactions involving proteins which are in the internal membrane. They're integral protein complexes in the inner membrane of the mitochondrion. There is what we call a term that I'm going to use today and also in terms of photosynthesis, coupled electron transport and ATP synthesis. That is, as electrons are passing through various carriers, ATP is being synthesized. This coupling is absolutely critical to what's going on in the mitochondrion. There is a large delta G for this reaction. That is the reaction, the reoxidation of these compounds, NADH, NADH, and FAD2. A large negative free energy change. Every time you oxidize one NADH, to give you an idea, plus oxygen to NAD plus water, the free energy change is minus 53 kilocalories. So you can see where most of the energy that was available in glucose is. We have 6 NADHs here. We have 2 NADHs there. We have FADH2, which is has slightly less energy. Most of the energy that was in the glucose molecule is now stored in basically these compounds. And the trick the cell has to do is it has to not only oxidize these compounds, but it also has to convert as much energy as it can into ATP. And that's what we're going to be talking about today. I actually brought a number of wonderful figures to show you, but I don't think this thing is working. Okay, I've got to introduce some of the carriers, some of the cast of characters. What I've said is we have the inner mitochondrial membrane. Here it is. This is the inner mitochondrial membrane. So there's another membrane out here. Outer. Inner. And we have a series of complexes, protein complexes. And they are conveniently named 1, 2, 3, and 4. Brilliant. And these 4 complexes interact with NADH and FADH2 and pass electrons through a number of electron carriers. Ultimately, those electrons end up in oxygen. What are the kinds of carriers I'm going to be talking about? The first ones, which are historically the most significant, are compounds which are known as cytochromes. What are cytochromes? Cytochromes are electron transfer proteins. They transfer electrons. They contain heme. Remember heme from hemoglobin. When I talked about hemoglobin, I said hemoglobin has a heme in it. Well, cytochromes have a heme in it. The difference between hemoglobin and a cytochrome is the cytochrome, the heme group, the iron in the heme, in the middle of this ring, does not get oxidized in hemoglobin and reduced. It carries oxygen around. The iron in cytochromes gets oxidized and reduced. So it goes from Fe+3 state to an Fe+2 state. An electron plus Fe+3, and then that Fe2 is reoxidized. So there's an oxidation reduction process involving the iron of the heme group. The heme group is a complicated, don't even copy this down, this part down. It's too complicated. It is given schematically in figure 110, but this is not something you have to know, the structure of heme. You can see there are 4 rings, nitrogen-containing rings. The iron sits in the middle. The heme group has various side chains on these rings, and this leads to the result that there are various kinds of cytochromes in mitochondria. There is one cytochrome known as cytochrome A. It has heme A, where this side chain is modified. There's a cytochrome C, where this side chain is modified. The modifications are unimportant. The chemistry is unimportant. What you have to know is that there are 3 cytochromes known as A, B, and C in mitochondrial membranes in these complexes, and I'll tell you where they are. It's very interesting to talk about a British biochemist, David Kalin, working in the 1920s, working with a very sophisticated instrument. This is it. This is a hand spectroscope, and for those of you that come to office hours after class, you can look through this and see what David Kalin saw. This is what he was using to look at living tissues, like cells. He studied moth wings because they were very thin, and he could see things, absorption bands of things that were changing. For example, now we know that cytochrome A is associated with a 605 nanometer absorption band, B 560, C 550. He saw these absorption changes with a hand spectroscope, and he figured out, using this very sophisticated instrumentation, how the cytochromes function in respiration. I think there's a lesson here. You don't need $1 million machine to do measurements if you've got good ideas. So here it is. And the idea that emerged from Kalin's work was that there was an electron transfer from a B cytochrome to a C cytochrome to cytochrome A. It's more complicated than that, but it is basically correct. He worked out the basic principles of electron transfer through the mitochondrial electron transport chain. Now, this was a very important piece of work. It obviously, I think, no question deserved the Nobel Prize, but there was a competitor of his, very powerful scientist in Germany. Otto Warburg was his name. And he essentially didn't believe this. And he prevented Kalin from ever getting a Nobel Prize. He just stopped. He had a Nobel Prize, and he was a very prominent German professor, didn't wear plaid shirts to lecture or thing. Probably didn't lecture anyway. But he was pretty important, pretty powerful, and he influenced science for a number of years. Some of his ideas were right, some of his ideas were wrong, okay? Okay, what happens? Second important carrier that I have to introduce to you is a compound known as ubiquinone. This is ubiquinone, this is ubiquinol, this is the oxidized form, this is the reduced form. What happens to, firstly, the structure of ubiquinone? It's an aromatic ring with a very long tail. I didn't draw the tail on this compound. It has 20 carbons or 25 carbons on this tail. This is a super hydrophobic molecule. Okay, very important. Hydrophobic, where would you expect to find it? Obviously in a membrane, and it is in this mitochondrial inner membrane. Ubiquinone can be reduced, and when it is reduced, it picks up 2 protons and 2 electrons. So this is different than a cytochrome where you're simply transferring electrons. But again, this compound is in the membrane. It's not bound to a protein. Oh, I should mention something about the cytochromes. And it is freely mobile in the membrane. It moves. I forgot to mention that not only are the cytochromes A, B, and C, have different absorption properties, they're different proteins. Actually, there are two kinds of cytochrome C. There is cytochrome C and cytochrome C1. And these represent different proteins in the mitochondrial membrane. Cytochrome C1. And we'll talk about both of them. So there are really 4 cytochromes we have to be worried about. When you look at these 4 complexes, they are essentially functioning as enzymes. Each one of them carries out an enzymatic reaction. And what's shown in figure 112 is the overall scheme of electron transfer, so that you can see in figure 112, NADH is interacting with complex 1. Where's the NADH? This is the matrix. Is the NADH in here or is it in here? It's in the matrix. Why is it in the matrix? Because that's where the Krebs cycle is. Krebs cycle makes NADH. So NADH is converted to NAD by complex 1. And there are electrons that go through complex 1. Complex 1 does not contain cytochromes. Those electrons are transferred to UQ, this compound. And that's, I believe, shown in figure 112. Well, there are no arrows, so you can look at 112 where there are no arrows, or you can look at 113, which is a fuller representation of this electron transfer chain. Maybe 113 is a little better, okay? So the electrons are being passed from NADH through this complex, and they will go to ubiquinone. The ubiquinone then is reduced. It takes 2 electrons. It takes 2 protons. Complex 2 is actually the succinic dehydrogenase of the Krebs cycle. And what it does is it oxidizes succinate, and it produces FADH2. So this is where the FADH2 product is produced. The electrons in FADH2 are transferred again. I'll put it here, FADH2. This is actually in the enzyme, but don't worry about it. Are transferred to ubiquinone. So ubiquinone in the mitochondrial inner membrane can receive electrons either from NADH or FADH2. Sort of two pathways going to the same compound. Ubiquinone then passes electrons to complex 3, and complex 3 is where, for the first time, we see cytochromes. We see cytochrome C1 in complex 3, and we see the B-type cytochrome in complex 3. So there's a B cytochrome and a C cytochrome, and the electron actually passes between the B and C cytochrome. Not a big deal, but the electron ends up going through this complex, and it ends up in a protein which is not a membrane protein. This is cytochrome C, not C1. This is C1, and this is C. Cytochrome C is a fascinating protein. The actual structure of cytochrome C is shown in figure 111. It's a little itty-bitty protein, relatively speaking. You can look at the ribbon diagram that shows you the peptide chain, and if you look closely, it's hard to see. You're looking in a crevice. There is a heme group, and there's a little round ball. That's the iron. Now, cytochrome C is a peripheral protein. So it can be readily removed from the membrane by usually just washing with, I think, high concentrations of salt. And that makes it very accessible to examination by techniques such as crystallization and X-ray diffraction. And there are lots and lots of cytochrome C molecules for which we have structures. It's been a very useful molecule in studying the evolution of organisms and systems like this. So the electrons are passed from ubiquinol through complex 3 to cytochrome C, and then they are passed through complex 4, which is functioning as a cytochrome oxidase. Again, remember, I haven't drawn oxygen on the board. And when electrons pass from cytochrome C through cytochrome oxidase, they are transferred to oxygen, molecular oxygen. So cytochrome oxidase is called the terminal oxidase of the respiratory chain. It's the only place where oxygen directly interacts with some of these components. There is in complex 4, there are 2 A cytochromes, A1. See, I'm running out of room. This is 4. There's an A1. There's an A3. And there's also copper. So cytochrome oxidase is a very, very complicated enzyme, very interesting enzyme. Cytochrome A3 is the component that actually binds oxygen and reduces it to water. It is also the place where respiratory inhibitors, like cyanide, which I mentioned when I was talking about inhibitors, reacts. You die from cyanide poisoning because cytochrome A3 binds cyanide, and you have no respiration. Oxygen cannot react with cytochrome oxidase.
Is there a Anaero cytochrome below A3?
That's copper.
Copper.
It's copper. Yeah, there are actually 2. This molecule has 2 cytochromes. A and A3 are slightly different, not important. There's copper in there, and all of these compounds, all of these metal compounds, are there to carry out a rather complicated reaction, which is convert oxygen to water. O2 to H2O. Mechanistically, very difficult to do. Okay, so we've got these 4 electron transfer complexes, and as I said, you should think of them essentially as enzymes. This first complex oxidizes NADH and reduces ubiquinone. The second enzyme oxidizes FADH2 or succinate, doesn't matter, reduces ubiquinone. The third complex oxidizes ubiquinol and reduces cytochrome C. And the fourth complex oxidizes cytochrome C and reduces oxygen to water. Everybody okay with that? Yes? Yeah, they are enzymes. I mean, they are enzymes, but they work together. They work together. There's no, you never find a complex 3 by itself, and you never find cytochrome oxidase in any other place other than this electron transport chain. So I think it's easier to understand if you say, well, I've got these 4 complexes, and each one is carrying out an enzymatic reaction. The product of the reaction is used by the next complex. Okay. Okay. What have we done here? We have finally involved oxygen in the process. We have oxidized our reduced NADH and our reduced FADH2. So that part of the system is done. But what we still have to deal with is ATP. And remember, 38 ATP. When people were studying oxidative phosphorylation in the 1950s, the biochemistry of this pathway, that is, people were isolating these complexes and studying what they did. The characterization of the electron transport chain was moving along quite well, and they were understanding it. The problem ultimately came, how do mitochondria make ATP? Well, the only system at that time for which we understood how ATP was being synthesized was the reaction I talked about last time in lecture, the glyceraldehyde 3-phosphate dehydrogenase. That reaction, there was an oxidation, a high energy, a stable chemical compound that had a phosphate on it that was an energetically rich compound was formed, and that phosphate could be transferred to ADP. So people spent a lot of time looking for compounds like 1,3-diphosphoglyceric acid somewhere in this process, because that was the model that was used to study ATP synthesis. And they spent a lot of time, and it turns out it was all wasted. It's not a nice thing to say, but they couldn't find anything. And they kept saying, well, we can't find it because it's very unstable. So they tried other methods, and nobody ever found a phosphorylated intermediate involved in this pathway. And finally, in the early 1960s, I put this name on the board, Peter Mitchell, down here. Peter Mitchell, a very interesting man, working in England, came up with a totally different model. And I like to talk about Peter Mitchell because his career is so atypical of the kind of academic career that we're used to. He was trained as a biophysicist. He worked on ion transport in various systems, but he never really worked at a university. I think he worked for Guinness Brewery for a while, making beer, I guess, or drinking beer. And he may have been at Cambridge for a while, but not in an academic position. And ultimately, or finally, what he did was I think he was living in northern England. He moved to western England, Cornwall, which is 3 hours away from London. Nobody ever goes out there. It's a beautiful place, but nobody might want to live out there. And he had a house there, and his house had a laboratory in it. And he did experiments in his house. And he came up with this idea that the reason why people couldn't find this phosphorylated intermediate is because it didn't exist. And he came up with a different idea, a different model, known as the chemiosmotic mechanism for how mitochondria make ATP. And there are 3 or 4 points about this model that you have to be aware of. The important points concerning the chemiosmotic model. Number 1 is the membrane is impermeable to protons. Well, you understand enough about the structure of a biological membrane to realize that protons will not cross this membrane very readily. The second important feature of the model is he proposed that as electrons are moving through all of these carriers, and you can look at figure 113 for this, there is a movement of protons across the membrane, such that you end up with a gradient of protons, high on one side of the membrane, low on the other side of the membrane, and that's what's shown in figure 114. As electrons are moving through the electron carriers, protons are moving into the matrix. I hope I get this right, because I often get it wrong. No, that's wrong. They're not moving into the matrix. Back off on that. They're moving into the intermembrane space. That's what's shown in figure 113. They're moving from the matrix to the intermembrane space. So we have proton movement across the membrane like this from here. And you have proton movement across the membrane at various places during the electron transfer process. I'm not going to talk at all about how protons move across the membrane. This is a complicated story, and there's not enough time, but this is his idea, that a proton gradient was established across the membrane. The gradient of protons is a source of energy, and those protons want to come back across the membrane, right? If you end up with a lot of protons up here and very little protons in the matrix, there's a force to drive those protons back across the membrane. And he proposed that there was an enzyme in the membrane, which could carry these protons back across the membrane, and that would release energy, and that energy would be used to make ATP. This complex is known as, there's the protein, an ATP synthase, sometimes said as an ATP synthetase. I use those terms interchangeably, but I'll try to always say ATP synthase. So these were the basic principles of the chemiosmotic mechanism. 1, no permeability of the membrane. 2, electron transport. As electrons are moved, proton gradient, protons are moved. The proton gradient is an energetic, it's a storage mechanism for energy. And if you then have an enzyme that allows these protons to go back across the membrane from the intermembrane space to the matrix, this can result in the synthesis of ATP. Now, look at this mechanism and think about what I just said about substrate level phosphorylation. In substrate level phosphorylation, you form a phosphorylated intermediate, which can be converted to ATP. In the mitochondrial system, you don't form any phosphorylated intermediate. You form a proton gradient. And that is the driving force for ATP synthesis. So, of course, it's not very easy to find a gradient. You can't isolate a gradient. So it was something new and different. When Mitchell proposed this idea at a conference, I don't know, the mid-1970s or 80s, early 80s, it was reported, I mean, I know people who were at that conference, and people, they looked at him like he was crazy. And basically they said, go home, go back to your country estate in England, because this doesn't make any sense to us. And it didn't make any sense because nobody was thinking along these lines. So a good model is a model that can be tested. This model was good in terms of testing the various parts to it. For example, are there protons that are moving when electrons are being transferred? That is this area, the intermembrane space, becoming more acidic and the matrix becoming less acidic? Well, that's something that actually can be measured. It's not easy to measure, but it can be measured, and people started measuring pH changes accompanying electron transport. And yes, indeed, there were pH changes accompanying electron transport. And then the enzyme, the ATP synthase enzyme, which was predicted to be able to use this gradient, was known in a different context, but it was known that this was an enzyme. There was an enzyme on this membrane that could potentially make ATP, and that was studied, and it was shown that it worked as well. And there were a couple of experiments that were done with the photosynthetic system that I think are even more compelling in terms of the chemiosmotic hypothesis that I'll talk about next time. But slowly, support for the model built so that we had, let's say, 1960. There was, this is year. Actually, this was published, and I can't find it. Although I would love to find it again. Believers, okay, on that axis. And in 1960, there was one, and that was Mitchell. And then, very slowly, it went up, it went up, it went up, it went up like that. And about, I don't remember when he got the Nobel Prize, 1986 or something like that. He was given the Nobel Prize in Chemistry for elucidating the mechanism by which mitochondria and, I'll say now, chloroplasts make ATP. Very, very important, obviously very important process. This enzyme, the ATP synthase, is a very interesting enzyme. It sits in the membrane. You've got to look at figure 49. Figure 49 is a complicated picture. Just look at the model in part F. Well, page 49. I'm sorry, page 49. There are two parts to this enzyme. They are known as the enzyme historically. I'll draw it again. This, you don't have to remember that. Here's the membrane. Here's the enzyme sitting like this. There are two portions. There's a membrane portion, which is known as FO, and there's a sort of extrinsic portion, which sits on the membrane, which is known as F1. F1 is this knobby thing that you see in figure, it's figure 115. It's got a number of different subunits sitting out there, and this is the part of the enzyme that actually makes ATP from ADP and phosphate. And this part of the enzyme is a region where protons come across the membrane. It's a very interesting region. It's made up of a number of very hydrophobic proteins. You can see there are they almost look like tubes. And here's a case where you've got a very, very hydrophobic group of proteins. There are, I think, 9 of these subunits in the membrane. And in the middle of the membrane sits an aspartic acid, which is clearly doing something. You don't put an aspartic acid into a membrane just for laughs, and it is clearly involved in moving the protons across the membrane. It's possible to take this part of the enzyme, the F1 part, away off of the membrane because it's not exactly peripheral, but it's pretty much peripheral, in which case it becomes an ATPase, which is something that Mitchell proposed. It can hydrolyze ATP to ADP and phosphate when it's removed from the membrane. You see that there is a part of the protein that's in the membrane that kind of reaches up to the F1 part. The mechanism of this protein is actually fascinating. There's something like this. And there is evidence for this mechanism that this, as the protons come through the membrane portion of the protein, the top part spins. And that results in conformational changes in that portion of the molecule that make ATP. So you now see this enzyme being called a molecular motor, new term, because it is actually spinning around, and the spinning has been shown experimentally. The other point about Mitchell's idea was it was known that there were a number of compounds which, when added to mitochondria, allowed electron transport to occur, but no ATP synthesis occurred. These compounds are called uncouplers. Because I mentioned that the two processes of electron transport and ATP synthesis are coupled. That it means they go together. And you can see why they go together, because the electron transport chain is making the proton gradient exist. But you can find compounds, and there were a variety of compounds. They had nothing in common with each other, apparently, which, if added to mitochondria, they would allow electron transport to occur, but no ATP was made. And Mitchell looked at these compounds, and he said, these are compounds, uncoupling agents are compounds which bind protons. So what happens when you add these uncouplers is they go into this space primarily in this case, and they will bind up the protons and take them away, make them unavailable for the ATP synthase, but the electron transport chain now can still do its thing because there's no pressure to stop. As you build up the gradient of protons, this will put a back pressure on electron transport, and it will slow down. You get rid of this back pressure by, one, allowing the protons to come across the membrane and make ATP. But an uncoupler removes the protons as well in a useless manner. So it made a lot of sense in terms of chemiosmosis that uncoupling agents were stopping protons moving across the membrane, but allowing electron transport to occur you will use an uncoupling agent next week in your photosynthesis experiment. So what we have to do now is we have to look at how much ATP is actually made. Figure 116. There's some disagreement among people as to how much ATP is actually made, but it's not significant. I think the differences are minor, okay? Basically, the classical view is that every time you oxidized an NADH to oxygen, you made 3 ATPs. Every time you oxidized an FADH2 with oxygen, you made 2 ATPs. And for the sake of the arguments and just simplicity, this is the way I've always talked about it, these are the numbers that I will use, and they're used in table 116. So every time you have, well, actually the simplest way to do this is to go back to figure 109, the output of aerobic metabolism. It's very simple. You have 4 ATPs, which are made by substrate level phosphorylation. You have 10 NADHs, which are present from glycolysis, pyruvate oxidation, and the Krebs cycle. Those 10 NADHs, when oxidized, will produce 30 ATPs. You have 2 FADH2s, when oxidized with molecular oxygen, will produce 4 ATPs, 34, and you have 4 ATPs by substrate level phosphorylation. Okay, I had those on the board. 2, 4, so 4 substrate level phosphorylation, giving you 4 ATPs for a total of 38 ATPs. That's where this number comes from. And as I said, I think Russell has a slightly different number because some people feel this number of 3 ATPs is a little squishy. Maybe it's 2.5, or maybe it can change under various conditions. The difference, I think, is it's not that important. The point here is you're making 19 times more ATP than you are during fermentation, where you make 2. So respiration is enormously more productive, and that's because you're carrying out a complete oxidation of glucose. Much more energy is available, and that a lot of that energy can be converted into ATP. So that's why it's very important. All of these wonderful figures I brought. Can't use them. Terrible. One of the things that you have to do is you have to calculate the efficiency of respiration. Now, it turns out that this number of 38 ATPs is what one would expect to see in a prokaryotic cell. In a eukaryotic cell, this number becomes 36 ATP. Why does it become 36 ATP? So this is 38, and this is in a prokaryote, because it becomes 36 in a eukaryote for a simple reason, and that is 2 of the ATPs in a eukaryotic cell are in the cytosol. 2 of the NADHs in a eukaryotic cell are in the cytosol. For them to undergo oxidation by the respiratory chain in the inner mitochondrial membrane, they have to be brought into the membrane, into the matrix, and that costs the cell 2 ATPs. That is an ATP, it's an energy-dependent process. So instead of 38, your number in eukaryotes becomes 36. To calculate the efficiency, you would say, I have 36 ATPs. Each one has 7.3 kilocalories of energy stored, giving me 263 kilocalories of energy. 263. I started with how much energy in the glucose molecule? 686. Remember 686. So to calculate the efficiency, you take 263 over 686. You multiply that by 100, and you get 38% or something. 38%, right. 38% efficient. 38% of the energy available in a glucose molecule is now present in ATP. Well, somebody's going to say, well, that's not very good. 38%. Actually, it is very good. When you burn gasoline in a car, the efficiency of that process is 25%, 20% or something. 38% is good. Let's put it that way. Where's the rest of the energy? Where'd it go? Heat. It's gone. What I think you should appreciate is how complicated this series of reactions is in respiration. You have basically 10 reactions in glycolysis, and you have 10 reactions in the Krebs cycle, and then you can't even count the number of reactions here. They're not countable because we don't really understand how NADH is oxidized by complex 1. We don't understand how this complex 3 works, but you probably have in total 50, 60, 70 reactions. There is a model for how ATP is actually made by the ATP synthase. It's a rather complicated model. So we don't know. We can't say how many steps are there in the process. And each one of them has its losses, and you end up with 38%. Now, I expect you to be able to calculate the efficiency of ATP production for various things that you might encounter a week from Friday. So you should feel comfortable with this, how I did it. And it's just taking the amount of ATP you've made and calculating how much energy is in those molecules. And then you have to know the total starting amount of energy. And you get an efficiency, okay? Okay, I have a couple minutes. 2 minutes. 2 minutes, that's good. Where are we making ATP? Where is ATP actually being synthesized by a mitochondria? No. The enzymes are in the inner membrane. Where does ATP end up? What's this space down here? It's the matrix. ATP is in the matrix. And that's kind of a silly place for there to be ATP because all the metabolism is happening not in the matrix. So what you need to have and what exists are transport systems for taking ATP across the inner mitochondrial membrane into the intermembrane space, and then across that membrane to get it into the cytosol where it's needed. So, having ATP synthesis produce ATP in the matrix is unfortunate, but that's the way it is. It's got to do with the direction the gradient is made and the orientation of this ATP synthase. This is the acidic pH, and the ATP synthesis enzyme is on the other side of the membrane, so you end up with ATP in the matrix. Okay, I'll stop there. I finished. I got through it all. And I'll see you all on Friday.